Our names are Maria Alejandra Vargas and Daniel Valenzuela, and we are going to present cell recognitions. Basics of cells. Cells are the basic building blocks of all living things. Cells respond to the environment around them. Cells are surrounded by a cell membrane that maintain the physical integrity and control the movements of particles. This cell membrane is composed primarily by proteins and lipids. It also has some elements which are glycolipids and glycoproteins, which maintain the stability of the membrane and facilitate cell recognition. What is cell recognition? Cell recognition is the process in which a cell recognizes or identifies its environment. It's one of the ways in which cells communicate with one another. It is important when building an organ system or fighting a foreign invader like viral or bacterial infections. Cell recognition has two very important elements, which are glycoproteins and glycolipids.
Now, to determine what is the relationship between glycolipids and glycoproteins in order to use them in the process of cell recognition, we need to determine what are the both of them. So, what are glycolipids? A glycolipid is a type of lipid with carbohydrate group attached to them by a glycosylic bond. Their main functions are maintain the stability of the membrane and attach cells to one another to form tissues. They are found on the surface of all eukaryotic cell membranes. And there are three types of glycolipids: glycoceroglipids, glycosphingolipids, and glycophosphatidylatinositols. Now, what are glycoproteins? Glycoproteins are simply proteins with a sugar attached to them. In other words, are proteins that contain oligosaccharide chains covalently attached to polypeptide side chains. Glycoproteins are involved in nearly every process in cells. They have diverse functions such as in our immune system, protection of our body, communication between cells, and our reproductive system. There are two common types of proteins: N-glycosylation and O-glycosylation. So now, how do glycolipids and glycoproteins help in the process of cell recognition? Basically, cells recognize each other by binding to surface molecules, often carbohydrates, on the plasma membrane. Some membrane carbohydrates are then attached to lipids, creating glycolipids. Also, there are some covalently bonded with proteins to create glycoproteins. Since they are now linked to the interior side of the cell membrane, they can now interact with the surface molecules of other cells, which then, through this, scientists are able to use this characteristic in cells in order to recognize them. Thank you very much.